In Chapter 7, we'll look at how characteristics are inherited using Mendelian inheritance. First of all, we need to look at kind of some basic terminology, and you're already familiar with some of these. You know about chromosomes because we covered them in Chapter 6, and genes are inherited on chromosomes. And we've looked at some of these, so when we talked about the chromosomes from parents, so from the mother and the father, and particular locations on the chromosome. I know in Chapter 6 we talked about eye color, so maybe at that location on the chromosome it contains information for eye color. Those bits of information are called genes, and different forms of a gene are alleles. So having a blue gene and a green gene, those are different alleles of eye color. So same gene but different alleles because they are controlling different colors. And the way characteristics are inherited, your parents each have 2 chromosomes. When they produce either egg or sperm using meiosis, they separate those chromosomes, so they end up with 1 chromosome of each type in the egg and sperm, and those are combined during fertilization to give 2 to the offspring, 1 from the mom and 1 from the dad. And so all of that should be review from the previous chapters. In addition to basic traits and characteristics like eye color, some traits or characteristics you probably wouldn't want to inherit. This is an example. In the parents' chromosomes, they have a normal gene and they have a defective gene, or a gene that has a problem. And in this case, that defective gene affects a pathway in metabolism. So normally metabolism, you take a product, something that you eat, it's transformed or changed by an enzyme into something else. So it takes our substrate, the enzyme changes it into a product, and then you have a second enzyme that will change it again, and oftentimes this can go in many, many steps. Maybe you have a third enzyme, and then maybe this is just waste product. Well, in this particular case, it's a normal part of metabolism that you have enzyme 1, enzyme 2, enzyme 3. One of those enzymes isn't working correctly. So maybe enzyme 1 is just fine, enzyme 2 is fine, but 3 has a problem in the DNA, and that's represented by that point in the chromosome. So if enzyme 3 isn't working, this builds up in the body instead of being changed to waste. In fish odor syndrome, this molecule building up in the body just happens to smell like fish. So if a person has a normal chromosome and a defective one, the normal one will make up for it. So this one will make enzyme 3 and it'll be just fine. So the mother smells normal. The father happens to have that same problem and a normal chromosome, so he looks normal too. However, if the mother passes on an egg containing the dysfunctional or defective gene, and the father passes on a sperm with that defective gene, and the baby ends up with 2 copies of the problem gene, then the baby will not be able to carry out this process, changing this to a waste product that doesn't have an odor. And normally with fish odor syndrome, it's not noticed when they're babies. However, at adolescence, kind of soon after puberty, you start noticing that these genes are not working, and so this smelly molecule builds up in the body. It has nothing to do with them showering, so they might shower and scrub their whole body. As soon as they step out of the shower, they still smell like fish because it's inside. So that molecule comes out in their sweat, in their saliva, in their tears, so everything just smells like fish. And people with the syndrome don't notice their own smell, but other people might notice. There's a classic example of kind of one of the first cases where fish odor syndrome was noticed, and it was a gentleman, and he had grown up normally and became a police officer. And he started having like suspects in the interrogation room complaining that it smelled like fish. You know, it smells like fish in the room, and he didn't know what they were talking about. And then he had his coworkers start telling him, hey man, you need to use a different gel in your hair because man, that stuff smells like fish. And he couldn't smell it. He didn't notice it because he was around it all the time. And eventually figured out that he had 2 copies of this defective gene. So he smelled like fish. So that's one thing you don't want genetics to give you. So the way genetics works, it uses heredity, so that's passing characteristics from parents to offspring. And humans have actually been looking at heredity for thousands of years and using it very effectively. So breeding agricultural plants and animals and all of our pets and things like that, they have been bred just by looking at the parents and crossing parents that have the characteristics or traits that you want in the offspring. However, the actual mechanism or how that worked was only understood recently, in the last 100 years or less. And since we've started understanding how heredity actually works, we found in humans there are over 9,000 single gene traits. Some examples are cleft chin or not, attached or unattached earlobes, the widow's peak hairline or the straight, if you can roll your tongue or if you can't. And all of these characteristics, because they're single gene traits, they have a very simple pattern of inheritance. So if you have one of these traits, look in your family. Your family members should also have, some of them anyway, the same traits. Some other ideas before we really understood the mechanisms of how traits were passed from parents to offspring. About the time people started using microscopes, the idea of preformationism was very popular. And preformationism, this is an example, because they were starting to look at this little tiny microscopic world, they thought, well, there were just little teeny tiny people inside. This is showing a sperm cell with a little tiny person, or it could have been a little tiny person inside the egg cell. And that person, when the egg and sperm came together, just got bigger and bigger and bigger. And then you had kind of different groups that would argue about whether this itty-bitty teeny tiny person was inside the sperm cell or if this itty-bitty teeny tiny person was inside the egg cell. So there were the ovocysts and the spermacysts. And they would actually have debates on where that teeny tiny person would be found. After that idea was disproven, because nobody ever found a teeny tiny person inside an egg or a sperm cell, the idea of blending inheritance became popular and remained popular for a very long period of time. And blending inheritance, it was kind of body fluid or liquid, so like blending paint. So the characteristics from the mom and the characteristics from the dad would mix together very much like paint in the offspring. It wasn't until Gregor Mendel in the mid-1800s that we discovered that it actually is kind of discrete units, so not like paint or liquid, but slightly different. We'll cover that as we go through the chapter. And Mendel didn't do anything especially unique. Other people had crossed lots of plants, lots of animals, and tried to figure out what was going on, so his experiments were actually very similar to previous experiments. However, he was the first to apply the scientific method. So he made observations, he developed hypotheses, so what he thought would happen, made educated guesses on what was going to happen. He designed experiments, carried out the experiments, and analyzed the results, and then compared that to his original guess. Nobody else had ever done that before. Gregor Mendel was a very interesting person. He started out, his family was just basically peasant farmers. They farmed the land, but they didn't own it, so they had to pay most of their income to the person who actually owned the land. And to give him the most basic of education, reading, writing, basic math, his family really had to sacrifice. Some of his writings from when he was older, he talked about his childhood and how his sister would have to go literally without a winter coat and freeze all winter so that he could pay for his education, and his family had to go hungry for periods of time so that they could pay for his education. So they really struggled to get him the basic education. However, he excelled at his studies. He did a very good job. And the local monastery had noticed that he was an excellent student, and they offered to pay for him to go to the University of Vienna, which was an excellent opportunity So he went to the University of Vienna, and that's where he was first exposed to the scientific method, to math and statistics. And when he graduated, he wanted to become an instructor or teacher at the college. And to become a teacher at the college, they had to pass an oral examination. So he would sit in front of a panel of people who already worked there, and they would ask questions, and he would answer, and then they would decide if he could become a professor or not. And he actually failed that exam partly because they didn't like his ideas on how characteristics were inherited. Everybody knew it was paint-like in nature, this blending liquid inheritance, and Mendel suggested it might be otherwise. And so they just didn't like that, so they failed him from his exam. And so then he had a choice. He could either go back to the monastery, take his vows, and actually become a monk, or he could go back and join his family and farm the land. He chose to go back to the monastery, took his vows, became a monk, and he eventually had a very successful career. He started breeding pea plants, which we'll talk about in a minute, right away, and he proved his ideas of inheritance. And eventually he gave up his studies and gradually moved up the ranks in the monastery to become kind of the abbot or leader of the monastery, which was a powerful and prestigious position at the time. So his success factors, what did he do? Well, he focused on specific traits, which a lot of people who had done breeding before hadn't. So he would look at one characteristic at a time. So if he was looking at flowers, he would look at their color, purple or white, and ignore everything else. If he was looking at the peas, he'd look at just the shape and ignore everything else, or look only at the color. So one set of characteristics at a time. He also first established what's called true breeding lines. And true breeding means that if you have purple parents and you cross them, they always give purple offspring. Or if you have white parents and you cross them, they always give white offspring. So he would allow his peas to self-fertilize for several years until he got these true breeding lines for all of his characteristics. He chose the garden pea for several reasons. It's small, very easy to grow and work with. It has lots of offspring, so lots of peas in each pod so that you can count them and do some math and statistical analysis. They also self-fertilize, which is a handy characteristic because you always know who the parents are. They actually fertilize themselves before the flower opens. So if you want to cross pea plants, you actually have to cut open the flower with the scalpel and use tweezers to transfer pollen from one plant to another. So he always knew exactly which ones were the parents. And they had many traits, so lots of different characteristics to look at. Some basic terminology. He found that some traits were dominant. They would show up or hide other traits, and some traits were recessive. They would be hidden. An example of that is purple flowers. If a plant had purple flowers, it would hide a white flower characteristic. And he set up all of his crosses in a classic Mendelian cross. And we'll start with monohybrid, mono meaning one, hybrid meaning you're crossing them. So we're looking at kind of crossing one trait at a time. He used 3 generations, P generation, F1, and F2, and P generation, or the parental generation, those were the true breeding plants. And that's a characteristic of all P generations, that they're true breeding. And so he would take a purple plant and he would cross it with a white plant. And that was his P generation. What he found in the F1 generation is it would either be purple or white. So in this case, they were all purple. So it would hide that white color, and that would tell him that this purple was the dominant trait because it showed up. So we have the P generation, and this is the F1 generation. They would all be purple. In the F2 generation, he would get mostly purple and a few white ones would show up again. And he saw this pattern over and over and over again, whether it was flower color, seed shape, seed color, pod color, height of the plant. He saw the same pattern. When he crossed 2 true breeding, one of the characteristics would be dominant and show up in the F1, and then the F2, crossing two of these purple ones together, or letting it self-fertilize, in the F2 you'd get mostly purple, and some of those whites would show up again. And this is what it would look like in his experiment. So we can see we were crossing a couple of true breeding, purple and white flowers together. First generation we get all purple. We can self-fertilize or cross them. The next generation we get a couple of whites, and then mostly purple. Mendel's explanation for the patterns that he saw was that each parent carries a single set of instructions. He didn't use the term genes, but we know that that's what they are now, and that's a term that we'll use. So each parent has a single set of genes, and they pass that to the offspring in either the egg or the sperm, so that the offspring receives two copies or instructions of the genes. Now keep in mind that Mendel knew nothing of cell division. He may have never even seen chromosomes under the microscope go through mitosis or meiosis 1 or 2. So this was all figured out just based on the patterns that he saw, not with knowing what was going on in the DNA. So he decided that those observed traits depended on the copies of the gene. Now remember, during Mendel's time, blended inheritance was the common knowledge. So everybody believed it was blending, so a liquid substance. However, blending didn't work with Mendel's ideas because you can't separate the paint back out. You can't take this mixed paint and separate that back into the original. But in his F2 generations, he kept getting the original characteristics back. So a better analogy rather than blending or liquid inheritance would be kind of like mixing Legos. So each colored Lego would be a different trait or characteristic, and the colors from the parents are mixed in the offspring. However, because they're discrete blocks, you could actually pick them up and separate them back out by color, which is how it happens. Each of those genes on the chromosomes gets separated out as it's passed on to the next generation. And this gives us Mendel's law of segregation, or Mendel's first law. Different textbooks refer to it differently, so make sure that you know both of them. So Mendel's law of segregation is also the first law, and that just says that two genes in an individual will separate when they're passed to the offspring. And you guys are already familiar with this. So if we have a parent here with two genes, say white and purple, and another parent with white and purple, those chromosomes are going to separate. So the white and purple from this parent will go into a different egg or sperm. Same thing with this parent, and then they will randomly be combined. So you could get two whites, one from each parent. You could get a white and a purple, again, one from each parent. You could get a purple and a white, or you could get two purples. So you have four possibilities given the parents' original genes that they're carrying. So this goes back to chapter 6 with independent assortment and also random fertilization. So you should be fairly familiar with Mendel's first law. Some more terms. So homozygous, homo means the same. Zygous refers to when you have a sperm and an egg cell that come together, that very first cell is called a zygote before it starts dividing. So that homozygous is saying, well, they get the same copy of the gene in their zygote, their first cell. So if we're looking at our Lego analogy, they would both get, or they would get red from each parent. Hetero means other, again referring to the zygote with the zygous. You get two different traits. So you get a yellow Lego from one parent and a red Lego. This would be analogous to you get the blue eye color from mom and brown eye color from dad, and then your actual eye color is a combination of these.